The Biocides work was one of the first projects that was funded by the Centre. So this is the report back on 2 years of that work. So it was a collaboration between ESR, Cawthron and the University of Otago. So I'll be presenting some of the work that they carried out as well. So firstly, thanks to the Centre for funding this work and showing some patience with us. We carried out some work with the dairy and red meat industries and really want to thank the people in those industries for their generosity with their time and with their data in some cases. I'd also like to thank Global Proficiency. Global Proficiency are a company up north, and they're responsible for carrying out assessments of biocides in New Zealand. We also had a lot of help from them. So we sort of knew what we wanted to look at, but we didn't have a definition that encompassed it, so we made up our own definition. Disinfectants or sanitising compounds, including water disinfectants, that may be inadvertently present in foods. So they're not meant to be there, they're not intentionally added to foods, and their residues are not specifically regulated. Now that last part isn't universally true. Some of these residues are regulated in some places, but they're not internationally regulated. By volume, most of these compounds are acid and alkaline cleaners, and these things, once they've been used, you don't really have detectable residues, but there's a whole group of other compounds that have specific signatures that can be looked for in foods. So we tried to carry out quite a wide range of activities under this project. We looked at usage, and this is where we had a lot of cooperation from the dairy and red meat industries. And that usage included looking at some trends, where we'd come from and where we were going. We looked at the regulation of these compounds in New Zealand and in some key trading countries. We looked a little bit at the analytical methods, and we found there were satisfactory analytical methods available in New Zealand. Nothing sort of universal that you could go and ask a lab to analyse for biocides, but if you knew what you wanted to look at, there are methods available. We looked at what information there was on the residues through the food chain to see if they persisted, where they might occur in the food chain. Phil Bremer and Miranda Morosa carried out some work on consumer perceptions, which I'll talk to in a minute. And we looked a little bit at microbial resistance. Antimicrobial resistance is a big topic these days. It usually relates to antibiotics, but you can also get resistance to these biocides. So, first case study, teat disinfectants. These are pretty important compounds. They're used to treat the teats of dairy cattle to prevent mastitis. For many years, the disinfectants of choice were iodine-based disinfectants, iodophores. And these not only had benefits for the control of mastitis, but some of the iodine carried over into the human food supply. New Zealand is a naturally low environment for iodine, so this was an adventitious benefit to human nutrition from the carryover of this iodine. There was a downside. These iodophores contain surfactants, so this is to get the compounds to stick to the teats, to the udders. And these used to be typically things called alkyl phenol ethoxylates. Now, if you look over to the right of here, if you look at the top, that's a schematic of a 4-nonyl phenol ethoxylate, which looks harmless enough. But if you then look at how the tail of these compounds can conform, at the bottom, 4-nonyl phenol can adopt the configuration which is very close to the female steroid 17-beta estradiol. And nonyl phenol does exhibit estrogenic activity. Weakly, but it does exhibit it. And this quite rightly got a lot of people concerned. And what we saw is that over a 10-year period, there was a strong move to replace these compounds. By 2017, monitoring carried out by the dairy industry no longer detected these at all. So they did a great job of getting them out of the food chain. However, these compounds had to be replaced with something else, and we now see that teat disinfection in the dairy industry is about 50-50 still iodine, but iodine compounds not containing alcohol phenols, and the other half is chlorhexidine. Chlorhexidine is also used extensively in the human medical system as a sanitizer. And with the issue of antimicrobial resistance, there is a degree of nervousness in human medicine about this being used in animal production. Okay, quaternary ammonium compounds. These are very effective sanitizers. They bond to the outer membranes of both bacteria and viruses, upset the outer membrane so it becomes leaky and compromises the microorganisms in that way. In about 2012, the German Risk Assessment Institute carried out an assessment on the two most common groups of quaternary ammonium compounds. That's benzalkonium chloride and didecyl dimethyl ammonium chloride. The European Food Safety Authority looked at this assessment and they set MRLs for these compounds in 2014. Now, these compounds are not wildly toxic, but as soon as there was an MRL, there was a point of scrutiny. These are results from the National Chemical Contaminants Program carried out by the Ministry for Primary Industries. And we see in 2013-14, when the MRLs were first brought in by EFSA, about 80% of raw milk samples contained one or other of these compounds. By 2016, 2017, they were gone completely, not detectable. Monitoring carried out by the dairy industry shows much the same thing in processed products. So, although the biocide levels in New Zealand dairy products are generally quite low by international standards, the industry is quite sensitive to what happens in the regulatory systems in their markets. And New Zealand industry has done a very good job of removing these compounds that there was concern about. However, in some cases, as we see with the increased use of chlorhexidine, these replacements are not always without their issues. So now we move on to the consumer perception work. This work was led by Phil Bremer and Miranda Morosa with one of their students, Lei Kong, and Professor Eddie Fang, who talked to this conference last year. So they conducted 4 focus groups, 2 in New Zealand and 2 in China, because we wanted to get some comparison of whether there were differences in the perceptions of these 2 populations. The idea of biocides and the idea that they could be present in foods was presented to these focus groups, and then they were asked to express a level of concern. So we see in New Zealand on the left there that the majority of respondents were concerned about this news, and the concern was generally vocalised around the idea that they hadn't known that these things may be present in food, so immediately they were concerned. In China, the responses were a little bit more equivocal, almost similar numbers of people concerned and feeling neutral about it. And there's an interesting difference in attitude here that the Chinese respondents felt that if there really was a serious issue, that it probably would have been dealt with. So they're a little bit more trusting. The next step was to reframe the topic. So to introduce the idea that these compounds came with benefits, and then to talk about the risk level associated with these compounds, and finally scientific evidence, which was the risk-benefit balance that these compounds had, that the benefits outweighed the risks. So the issue was framed in each of these ways, and then people were asked to restate their level of concern. So if we see here, the 4 on the right are those where there was concern expressed, in the middle where they were unconcerned, and on the right where they were neutral. And we see for New Zealand that when the topic is framed in terms of the benefits of biocides, the level of concern goes down. So people are quite, they can see a benefit for health from these compounds. Unfortunately for us scientists, simply framing the issue in terms of risks or risk-benefit didn't really produce much difference in their perceptions. Slightly different situation in China. Once again, expressing the issue in terms of benefits produced a decrease in the level of concern. Also the scientific evidence approach, the risk-benefit, so they seem to be a little bit more trusting of science in China, and this reduced the level of concern. So in summary for this risk perception, consumer perception area of work, generally consumers had not considered the use of biocides in food processing. Obviously they knew that the environment had to be clean, but obviously they hadn't gone that extra step of thinking how the environment became clean. Upon considering biocide use, many of the consumers were concerned, and providing information in different ways could perturb that level of concern. Generally, positive information decreased the level of concern and negative information increased it, but not always. And as you saw, there were differences between the New Zealand situation and the Chinese situation. Food manufacturers should not rush, perhaps, to discuss the use of biocide with consumers, or I'd actually rephrase that to say they perhaps need to be careful about how they broach this topic. Different consumer groups responded to information about biocides in different ways, and this is the difference between New Zealand and China. So, antimicrobial resistance. As I mentioned, most of the discussion these days is about bacteria developing resistance to antibiotics, but they can also develop resistance to these biocides, to sanitizers. However, when you look at wild populations, so if you just take isolates from food processing environments, the prevalence of antimicrobial resistance is pretty rare. However, it can be induced, so if you expose bacteria to suboptimal concentrations of these biocides, they can slowly build up resistance. So this means if you have a food processing environment where people are not using best practices, then there is potential for resistance to be developed. However, once again, there's a rider on that. Commercial biocides tend to be combinations of chemicals, and there's little or no evidence that bacteria can develop resistance to these commercial products. And just talking about the difference between antibiotic resistance and biocide resistance, the mechanisms for biocide resistance tend to be very general. Either the bacteria kind of shut up shop so that these compounds can't get through the cell wall, or they increase the activity of things called efflux pumps to pump the things out of the cell. The downside, I guess, of the generality of these mechanisms means that bacteria can often develop resistance to quite a wide range of compounds. They're either stopping them all coming in or they're pumping them all out. And that's the end. Any questions?